All right, this is the schedule for this evening. First, I'll share on some introductions, public relation information, and then Dr. Spangler will share on the day in the life. And then we'll end on with a Q&A session as well as a brief poll at the end. So if you're not as familiar with the American Chemical Society, the ACS is the world's largest scientific community, and members have access to career development, networking, member groups and communities, conferences, funding and awards, and etc. Also, you have access to a variety of various ACS career development programs, such as the Chemical and Engineering Use Jobs Database, the ACS Career Pathway Workshop, which is a workshop that can help you to really discover which career path is the best for you, either working in government, industry, academia, etc. They do also have the ChemIDP, which is called the Individual Development Plans. It's something that can help you really to figure out which career path is best for you. The ACS does have free career consulting. You don't have to be a member of the American Chemical Society, and they're normally held virtual office hours every Thursday, the first Thursday of every month from 12 to 1:30 p.m. And some of the upcoming session includes careers in chemistry, entrepreneurship, is graduate school right for me, careers in government, etc. And this is an opportunity to learn about different topics, different professional programs, and also where to connect with fellow scientists in the community. You can also get involved in various member groups and communities, such as women chemists, minority affairs, younger chemists, etc. You can also participate in the ACS conferences, such as the national meetings. The next ACS fall meeting will be in San Francisco of this year. There's also the Green Chemistry and Engineering Conference, which will be in Long Beach, California in June. And also there's CERMAX, which is the Southeast Regional Meeting in North Carolina. So this program is being sponsored by the Younger Chemist community, and our vision is younger leaders transforming the world through chemistry. And our mission is to advocate for, develop, and support rising chemists to positively impact their careers, the ACS, and the future of chemistry. Our goals is to advocate for younger chemists needs, concern, and contribution. Also provide career support and for diverse opportunities and development, and also provide community to foster connections and really being able to meet younger chemists from around the world. There's different ways to get involved in the Younger Chemist Committee. If you do attend national meetings, you can attend National YCC Symposium. You can also get involved in the local section, Younger Chemist Committee, such as the Virginia section. And there's also awards that you can apply for as well. These are pictures showing some of the programs that we've done in the past through the Virginia Younger Chemist Committee. We did a cidery tour last summer. We also toured the Evonik Corporation, which is a German company in Richmond. We toured another company locally, Anton Par. We even toured Storm Brewery, which was really fun. And also we did other programs related to diversity and inclusion, as well as various career programs. So we're very active in the Virginia section, and our goal really is to provide programs to foster the development of students and young professionals in the chemical sciences. If you're interested in connecting with us, you can find us on social media, such as Instagram, Twitter, Facebook, LinkedIn, and that's your email. There's a QR code if you're interested in learning more and get involved. So feel free to scan the QR code. And so the Day in the Life series was recognized in 2021 with the ACS ChemLuminary Award for best activity or program stimulating member involvement. And this award was also shared with other local sections across the Eastern US. And we're also recognized last year in another ChemLuminary Award, Outstanding Continuing Public Relations or Communication Program of a local section. And so for today's webinar, during the presentation, please ask questions in the chat. And also after the presentation, it will be an open Q&A session. So you can unmute and if you want to ask question, or you can also use the chat function. Next, I'd like to introduce our speaker for this evening. Dr. Leah Spangler is an assistant professor of chemical and life science engineering at Virginia Commonwealth University. She obtained her PhD in chemical engineering from Lehigh University, where she studied the single enzyme biomineralization of semiconductor quantum dots for biomedical and energy application. Leah then continued her research career as a postdoctoral research associate in Professor Gregory's lab in the Department of Chemistry at Princeton University, where she studied the relationship between protein structure and optical properties of photosynthetic proteins, amyloid fibers, and de novo proteins. Leah's current research includes engineering de novo proteins with enzymatic activity for application in scalable biomaterial synthesis and using time-resolved spectroscopy to elucidate the photophysical mechanisms of newly discovered optical properties in supramolecular protein systems. So I guess if we would like to introduce our speaker.
Here. Sorry. Okay, can you all hear me?
Yeah, we're good.
You can see it. All right, great.
Yeah.
Thank you so much for the introduction, Julian, and I'm super excited to be here and talk with you all today. So that was such a great introduction that some of what I'm going to talk about might sound like repetitive, but that's okay. I'll go into more detail. Yeah, so as Julian said, I just started this August as an assistant professor here at Virginia Commonwealth University. So I'm going to talk today about what I do day to day. I'm going to talk a little bit about the research that we get to do in the group, which is really fun. One thing that I think you should know about me is that I don't really like to lecture. So even when I teach, I think some of my students are here, they'll tell you I'm constantly doing interactive things. So hopefully we can make today a little interactive. I know we're recording, so maybe we can use the chat feature, but I want to start with a little interactive activity. So I just was wondering, just based on your impression, what do you think a professor does in their day-to-day life? And then we'll just see some responses. So if you can type into the chat. Oh no, don't. Well, that was probably the most obvious one anyway. But what are some things that you think professors do? And then I'll look at the chat and kind of address them. So I'll give you a minute. Lesson plan, yeah, definitely. Yeah, absolutely. So I'll talk about this a lot. Mentoring researchers is one of my favorite things that I get to do as my job in this job. So yes. Yes, write grants. I'm also going to talk about that. So a very important part. Yep, grading, reading articles, very important, right? So staying up on the scientific literature, especially in my field. Yep, sometimes, yeah, labs, helping in the research lab and also sometimes teaching labs. Yeah. Yeah, and so here's, a lot of these things, yeah, teaching graduate students. So I think this is a great list. You guys clearly have a lot of ideas about what professors do, which is awesome, but I'm going to talk about all of them in more detail. But a lot of times we think professors mainly teach, which of course we do, and that's one of my favorite things to do in my job is teaching. But we also, do a lot of mentoring, and we also do a lot of research, right? So I have a research group and we're kind of focused on a specific area, but what's great about being a professor is I'm kind of also teaching other people how to do research as well. So that's something I'll talk about. And then one thing that I didn't see come up, but this is also part of being a professor is mentoring and also doing service, right? So we also want to think about how we can give back and maybe try to promote our field and get more students interested in our field, right? And mentor. And so that's also something that I'm really passionate about too. And so I just want to mention that is part of the equation of what professors do. So thanks so much for participating, and maybe we'll try to do a little bit more. So I'm gonna kind of start by talking about myself generally, and I already had a great introduction, so I don't need to go over the slide in very much detail. And then I'll talk about my research group, and then I'll kind of give an overview of how I got to where I am, in case any of you are interested potentially in being a professor someday. So I'll do a little bit of a geographical history, but all my degrees are in chemical engineering. But of course, you know, ACS has been a big part of my PhD career. So chemists and chemical engineers obviously have a lot of similar research interests. And I even did my postdoc in a chemistry department. And so that was really interesting to kind of see the difference. And my research interests really include proteins and protein materials. So that's kind of interesting, right? Because those are more biological based systems. So not technically in my initial degree field, but things that I'm personally interested in researching. And then also I'm really interested in sustainable energy and material synthesis. And so as you can see, my research interests kind of span a lot of different fields. And that's something personally that I like, working very interdisciplinary and kind of bringing a lot of things together. I think that's where really interesting and fun research happens. And I'm also really interested in teaching, of course, chemical engineering, but also writing in science and engineering. So writing is a very important skill for scientists and engineers to have. And so I don't know that there has been a lot of, you know, vocabulary around this. So that's something that I like to teach and kind of work on. And I'm always working on my writing as well. So just something that also that I like to do and I get to do as a professor. Okay, so I want to talk a little bit more about my research interests that I kind of mentioned. So I mentioned one thing that I'm really interested in is sustainable energy. And that's something that I've been interested in even since I was an undergraduate. And one thing that I'm currently interested about this in particular is that when we think of something like a solar cell, that's a sustainable energy technology, we think, wow, this is great. It's just sitting in a field, it's collecting light from the sun, you know, it's generating electricity basically with no fuel, and that's very green and sustainable. But one thing we don't really think about as much is what happens to the solar cells once we're done with them. So, you know, we're kind of in a phase where maybe we've all just installed solar cells and we haven't got to that point yet, but they do have a lifespan. So we're going to have to think about how can we kind of recollect all the materials that are in the solar cell. But then we also don't always think about how they're made either. So when you take silicon, for example, a silicon solar cell, you essentially have to melt silicon down to, you know, at about 1000 C or something like very high temperature and pull a single crystal right out of this molten silica. And that's actually a very energy intensive process, right? So I believe I read recently the payback period for making a solar cell is something like 10 years in terms of energy that it's generating versus energy that was need to make it. And so, you know, I think it's important to think about when we're using these sustainable energy technologies, which are great and really good at making electricity, we want to think about also, you know, when we manufacture them and what happens to them afterwards. And so one thing that I'm really interested in looking at is, can we start to think about other ways to make these materials or find alternative materials to put into solar cells, right, that are perhaps more sustainable or environmentally friendly? And so in particular, I think about using biology, and I'll explain how I got to that point later. But biology is a really great way to manufacture things sustainably because, you know, it's all around us. It's evolved to be in ambient conditions and water. And so it's inherently, you know, environmentally friendly and usually very scalable. And so the biological system that I'm particularly interested in are proteins. And so I know we probably have a mixture of backgrounds here, but proteins are really are the workers of our cells, right? So they do all the functions in our cells that keep us alive, keep us going. You know, if you think about plants, that proteins are what are doing photosynthesis, right? And so they're kind of guiding all these chemistries. And so, you know, they're very important, very functional, and that's helpful if we want to think about making materials. But the other thing that's really interesting about proteins is their function comes from their structure. So I kind of have an image here of how proteins fold, right? So proteins are essentially, they start as very long chains of amino acids. So you can think of it kind of like a polymer. But because the amino acids have certain chemical side groups, they will fold into a certain structure. So they can fold into a secondary structure, like an alpha helix. You see kind of this helical structure here, or a beta sheet. They tend to stack a little bit. And then from there, they also fold into tertiary structures. And this is really where their function comes from, especially in the case of enzymes that have an active site where they're doing a reaction. And sometimes you can have multiple proteins coming together. And so as an engineer, I see this as a really great opportunity. So if a protein has a function that comes from its structure, and the structure comes from this chain of amino acids, maybe we can start to think about how can we engineer or design a protein using this chain of amino acids to do a specific function that we want. And so a question that we talk about a lot in my research group is, how can we engineer proteins that have functionalities that perhaps would be useful if we want to start putting them into sustainable energy technologies that aren't necessarily biologically based, right? So what I mean is, if you have a protein that goes into a solar cell, nature's not going to evolve a protein that's perfect for a solar cell, right? Nature's going to evolve a protein that's good at photosynthesis or something like that. And so how can we start to engineer protein ourselves for another alternative purpose? And so this is my inspirational protein slide. So of course, nature is an expert at evolving proteins, and there are a lot of proteins to choose from of different, structures and shapes. And so I just want to caveat this by saying, of course, we're not going to look at every single protein. We're going to find proteins that are close to what we want or very easy to manipulate, right? And start there and see what we can do. But we have a lot of different options, and there's a lot of space that we can explore. So I think this is a really exciting place to be, and you can maybe imagine doing a lot of different things even beyond sustainable energy, but this is kind of where we're starting. So just to quickly summarize generally what the lab is working on right now, because we're kind of just getting started and up off the ground, is we're really thinking about designing proteins that can make materials sustainably. So we're sort of focused on using proteins as biocatalysts so we can make materials, so inorganic materials like those solar cell silicon materials I mentioned. That would be a cool thing to do. And we're also interested in seeing, can we start to use proteins as a material? So perhaps making proteins that can act as light absorbers in solar cells and kind of act as analogs to inorganic materials, and kind of presenting an alternative type of device that could be used for specific applications. And so this is kind of an overview of the projects I mentioned. Here's my super awesome group with my students, who I will mention a lot today. I'm really excited to have such an awesome group of students to work with. And so we're kind of working on a lot of different projects. So I'll talk a little bit about one of the projects we're working on, which is using an enzyme to make quantum dots. So I'll talk more about quantum dots later. But also we're starting to think about how can we use proteins to tune materials and make materials of different shapes or improve their properties. And then I have some students who are also working on looking at proteins and seeing what needs to happen in order for us to put these into devices. So looking at, can we put proteins into something like a solar cell, for example? What would that look like? And also some students who are working on, can we tailor proteins and their optical properties for a specific function? And so that might look like having a protein with a specific structure, or maybe putting a chromophore or something that can absorb light into a protein. And so we're kind of doing a lot of different things, and there's a lot of really interesting projects going on, and that's why I'm so happy to have so many motivated students. Okay, so that's generally what the group, what my group does, but I want to take a step back and kind of explain how I got here. So I think it might seem a little out of the blue to bring biology and sustainable energy together. And I think a lot of that comes from my research background that I had coming to this point. So I want to talk about that a little bit. So this kind of boils down to my academic journey and also how I got to be a professor. And so if that's something you're interested in, this is sort of one way that you can get there. We'll add there are multiple ways. But I started my research career at Drexel. So that's in Philadelphia. If you've been there, Drexel is right next to UPenn. It's a really pretty area. There's lots of college, basically 2 college campuses right next to each other. And this is a really important part of my academic journey, was doing undergraduate research. So that's really what got me started to love research. So when I started my undergrad, I had no idea that you could get a PhD. I had no idea that professors had PhDs. So if that's you, that's normal. I didn't know either. And so when I found that out and found out that I could do research, it totally changed what I thought my college experience would be. And at the time, I was still very interested in sustainable energy. So I had the great opportunity to work on extremely thin absorber solar cells, which are just a specific type of solar cell, and got to learn a lot about that. And then, I guess my picture showed up early, but that's okay. So then from there, I moved on to do my PhD once I realized that was a thing and realized that was something I was interested in. And this is really where I started to kind of do some interdisciplinary work. So I was really fortunate to actually have 2 PhDs PhD advisors. Most people only have one. So my first advisor was Steve McIntosh, and he's a material scientist, so really interested in organic materials, and that was very similar to what I did in my undergrad research. But then I also started working with Brian Berger, so they had this kind of collaborative project, and Brian does a lot of protein engineering. And so what this project was looking at was seeing, can we use proteins to make something that can be used for optical electronic, which is fancy for, you know, LEDs or solar cell applications. And so this was a really fun project and really informed kind of my future research trajectory. So I'll talk a little bit about that now. I hope I'm doing okay on time. Yeah, I'm doing okay. All right. So, oh, and Lehigh is located kind of in central PA. This map is not completely accurate, so please don't use it as a reference. But it's a really, Lehigh's in a really pretty area, lots of mountains. So highly encourage if you get a chance to go there to visit. Okay, so I want to start by talking about, for this project, what a quantum dot is, for those of you who might not be familiar. So quantum dots are essentially nanocrystals that are about 2 to 6 nanometers in diameter. It depends on the material, but because they have such a small size, they have a really unique optical property. And so basically what happens is their electronic structure exists somewhere between a molecule and a bulk semiconductor. And practically what that means is, as you change the size of the nanocrystal, you get a change in the optical properties. And so here I'm kind of showing the quintessential picture of a quantum dot. So we have a bunch of vials of quantum dots, and in each vial is a population of quantum dots that is a specific size. And as we kind of move over, we see that this fluorescence is changing in wavelength, and that corresponds exactly with the size of the nanocrystal. So this is a really cool and unique property that I think originally physicists found out, and now we make them chemically, and we want to use them for a lot of things. And so depending on the type of material that you use, you can reach different parts of the electromagnetic spectrum. So you can reach the UV or the near IR, and so they're great for a lot of applications. So if I have any biologists in the crowd, you know, quantum dots are great for cell labeling. So you can pick 2 different sizes of quantum dots that fluoresce a different color, label 2 different parts of the cell, and excite them at the same wavelength, and you have kind of this multicolor image in your fluorescence microscopy. Of course, you can also use them in things like solar cells, but I think one application that more people might be familiar with are quantum dot TVs. And so I don't know if anyone's fortunate enough to have a quantum dot TV. I'm not. If you are, you might be familiar or not. The reason might be is because they're actually pretty expensive. And the reason that quantum dot TVs are so expensive is actually because quantum dots are pretty expensive to make. So here's my chemistry slide for the ACS talk. But the way that quantum dots are usually made is they're made in a round bottom flask, and you add a metal precursor. In this case, we're making lead sulfide, so we have a sulfur precursor as well. And then really important, it's important to have a capping ligand, so that keeps the quantum dot stable. And then oftentimes these are made in organic solvents and under inert atmospheres and at high temperatures, because that temperature is what's initiating the reaction or helping to grow the quantum dots in size. And so this is really effective to make quantum dots, but when we start thinking as an engineer, we often think about making things at large scale, and this isn't as scalable of a process. First of all, it's batch to batch, so that's usually harder to scale. The high temperature also makes it energy intensive. And of course, we want to try to use maybe more environmentally friendly materials. And so in my PhD, we were really wanting to see if we could come up with an alternative route that kind of solved a lot of these problems. And so I'm going to talk about next a process called biomineralization that happens in nature that we can actually exploit to use to make quantum dots. And biomineralization is great because it occurs in ambient temperatures, conditions, and it occurs in water. And so it's inherently more scalable, and we believe it to be more economical. And so I just want to take a minute and talk about biomineralization. So if any of you have been lucky enough to go to the beach yet this year, or anytime in the past, you know, I'm sure you've picked up a seashell and held a seashell. So that's actually the product of biomineralization. And so this is just a general term for the process any living organism uses to make minerals. And we can kind of exploit that process and start to think about making other materials. So I'll really quickly say these are some of my favorite examples. Of course, seashells made out of calcium carbonate. So whatever little organism lived in there has proteins and biomolecules that help shape the shell. These are radiolarians. So this is actually an SEM or an electron microscopy image of their skeletal structures. And so they have these really intricate and I think beautiful skeletal structures and you know that they grow around. And then another one of my favorite examples are magnetotactic bacteria. So these are bacteria that have iron oxide crystals in them, and they actually use them to orient themselves to the Earth's magnetic field so they can do some navigation. So nature has come up with a lot of really cool ways to interface with inorganic materials and use them. And so we thought, well, let's see if we can kind of draw from this and make functional materials that maybe nature doesn't want, but we would find useful. And so what we were able to do in my PhD was figure out that we could synthesize quantum dots, like I just showed you, using a biological approach. And so we actually used this bacteria, which I'm showing here, and that we eventually figured out was excreting a certain enzyme that was able to catalyze this reaction to make quantum dots. And so I'm kind of showing here, this is essentially what happens is this enzyme is able to take L-cysteine, which is an amino acid, and make sulfur, and then that sulfur can react with cadmium or lead or whatever other element that reacts with sulfur, makes a metal chalcogide and makes a quantum dot. And so we see here, this is an absorbance curve, and this is sort of like a quantification of the color of the solution and shows that we're able to have control of the size. And so I always like to show TEM images whenever I talk about quantum dots, just so you can get an idea of really how small they are. So this is a transmission electron microscopy image, and each of these little dots that you see is actually an atom. And so we can see that they're very crystalline. And we also see they are really small, right? So these are about three nanometers. And then we actually made a lot of different types of quantum dots, which I'm not going to talk about, but we were able to use them also for things like splitting water and generating hydrogen. So really trying to keep with that theme of environmentally friendly applications. And these are also really ideal for biomedical applications because they're already capped with amino acids usually or DNA. And so they're really good for that type of application. Okay, so then really quickly, I'll just move on. So my next stop after my PhD was to do a postdoc. And I'm sure you've had maybe other people here talk about a postdoc, but it's a good thing to do to kind of further narrow your focus into something that you're really interested in and help you kind of bring things together to have your own kind of research direction that's different from your advisors. And so here I was mainly working with Dr. Greg Scholes, who's a physical chemist who does a lot of measuring how light and energy move within systems. But while I was there, I also met and started collaborating with Dr. Michael Hecht. And so I'll talk about later, is a protein person. So it was kind of nice to have that. And I'm saying this to be inspirational to you, that always talk to people. Networking is so important. So if I hadn't started talking to Michael Hecht, we were actually talking about a different project and then kind of fell into another one. And so I'll talk about that here in a second. But I encourage you to always be talking about your research and kind of looking for possible connections. So the reason that I went to Princeton and what I was mainly studying I wanted to really learn more about proteins in things like algae that are really good at harvesting light and kind of understanding how does a protein control how light is absorbed and where the energy goes. And so I'm not going to go into a lot of detail, but I was able to take a few strains of algae and kind of grow them under different light conditions. And that actually affected their proteins, and their proteins essentially were modified so that they could absorb light in a different region. So here I'm showing an absorbance curve again, which is monitoring where the protein can absorb light. And we kind of see this shift into the blue, as we say, as spectroscopists, where after growing under restricted light conditions, they're kind of adjusting their proteins to compensate. And I, of course, was really interested in seeing, well, how does that adjustment happen on the protein level? And so we were also able to do protein crystallography and get the protein structure and compare before and after. And what we found out is actually that the structure wasn't changing, but the algae were actually swapping out a chromophore within the protein structure to change the absorbance. And so that was a really interesting finding. And it's still interesting to figure out, like, how is that happening? You know, what is the algae, how is the algae, you know, physically able to do that? And of course, we can continue to look into those things. But, you know, from a protein level, this was really inspiring because it shows how you know, we do have some degree of control over the absorbance spectra, and maybe we can start to engineer that. And so I also want to briefly talk about, I mentioned while I was working on that project, I started talking with Michael Hecht. So this is a picture of his lab group, and what they work on are de novo proteins. And so this was a really interesting thing to me. So de novo proteins are not from nature, right? So they've not undergone any evolution. They're made artificially and following a very specific pattern of polar and non-polar amino acids. And so what this means is they're going to fold into a very specific structure because this polarity really dominates how they fold. So here I'm showing a very typical protein that they make in the HEC lab, and you can see the non-polar residues are all facing in and the polar residues are all facing out. And so that really helps us to keep the structure stable, but then we can combinatorially vary the amino acid sequence as long as we follow this motif. And so what the HEC lab does is they have these huge libraries of these kind of randomly generated proteins, and they screen them to see if they have any activity at all. But as an engineer, I looked at this and thought, wow, this is really interesting. Maybe we can start to design proteins. As long as we stick to this motif, they'll stay pretty stable. So I'll talk more about that a little bit at the end. But the reason that I started working with Michael is actually they found a de novo protein that they thought might be able to biomineralize quantum dots. And to kind of cut short the whole story, essentially we did figure out that this totally random protein that was not designed for any function was able to make quantum dots by doing the same reaction that we found with the natural system. And so this was really exciting. And now the group is working on figuring out, more mechanistically what's going on. Can we start to add or improve onto the function of this protein? Can we find other de novo proteins that can biomineralize materials and kind of extend and do even more types of materials besides just quantum dots? And so this was a really, really great collaboration and really kind of, gave me a lot of momentum as I started my own research group to look into these things. Okay, so I talked about kind of doing my postdoc. And so of course, now I'm here at Virginia Commonwealth University, and I already talked a little bit about my lab. Oh, so here's my pretty picture of Richmond that I like to show. It's wonderful to be here. And so hopefully I've shown you that my research journey really helped me figure out my own research interests and how they can connect and how we can connect kind of disparate fields together and start to really look at new and interesting research questions. So hopefully, that inspires you and gives you a kind of an insight into the types of things that I get to think about day to day. And so I want to talk just a little more discreetly about what do I actually do day to day. So I know this is supposed to be day in the life, so I left it to the end. But I get to do a lot of really awesome things. So in addition to research, I get to lead an amazing team of student researchers. So, I get to help them run experiments in lab and train them and kind of analyze data. And that's really a part that I enjoy almost more than anything. It's one of my favorite things. I also get to teach classes, which is awesome. I really have fun teaching class. I like to make them interactive. And then also mentoring students, I mentioned several times, kind of goes hand in hand with both research and teaching, right? Helping students, with their academic work and also their career and things like that as well. This one kind of snuck in early. This is my other things I do more related to research. Of course, as you guys said, I read a lot of papers, and I really enjoy learning about what other scientists are doing. One thing that we professors also have to do is we apply for funding, because we really need money in order to fund our students and also in order to get all of the cool equipment and reagents that we need. And so a big part of what I do is trying to explain to other people why what I'm working on is new and cool and interesting, and why it's worth kind of funding and looking into. And so that's something that, of course, I'm always working on, especially since I've just started. But it's a really important thing that all professors do for the most part. And then, of course, we need to disseminate our research as well. So all of the cool things we're doing, we want to write papers. We want to make sure other people see what we're doing. We want to present our research, right? So I think it's really important to, if you're going to do stuff in the lab, you want to let other people know about it so they can maybe learn from it and use it in their own work. And then, of course, networking with other scientists. So at events like this, and at national conferences, I went to ACS as a graduate student and got to meet a lot of great people there. Chemical engineers have AICHE, the American Institute for Chemical Engineering. That's a really great conference to meet other people and kind of exchange and talk about your research and learn lots of new things. And then, of course, I kind of have some more specific conferences that I get to go to and other networks. So the Protein Society, in my case, is a really good one. So to talk with other protein chemists, even outside of chemistry and chemical engineering, it kind of broadens the field. And so I'm really glad that you guys are doing events like this, and I encourage you to keep up with that throughout your career because it's a really great way to meet people and kind of continue to have new ideas. Okay, so I just want to end with what I think are the best parts of my job. So the best parts of my job, I mean, I really enjoy being a professor, but working with students, is really something I look forward to every day. So I get to meet with students and teach students. Doing research, of course, is fun. So, getting to think about questions that maybe no one has the answer to and trying to figure out how we can address those. One thing that I really like about my job is I get to kind of be creative. So I get to kind of come up with, kind of new or interesting things and try to think outside the box a little bit and how can we approach problem solving. And so I'm sure you get to do this in a lot of jobs, but I really appreciate that I get to do it in this job. I get to explore topics that I'm specifically interested in. So one thing that's great about having your own research group is you really get to decide, what are some things that we're interested in and what kind of questions can we ask and answer about those things. And so you have a lot of freedom in that aspect. And that's something that I really enjoy. So I really get to focus on kind of bringing these two fields together. And maybe I wouldn't have had that opportunity otherwise. And then another thing I really love is that I'm always learning. So every day I always learn something new. So another reason I love having students in my research group, they teach me something new every day, science and otherwise, lots of things. And so that's something that I really love about my job. And then also we just have a lot of fun. So I think it's doing research is fun, learning is fun, and teaching is fun. So maybe that's just my personal opinion, but that's, I think, one of the best parts of my job is kind of being able to have fun and still joke around and still make great progress in your research. Okay, so I didn't make it very interactive, but thank you so much for your attention. I want to again thank the Virginia ACS chapter for inviting me to talk, and please keep in touch. Feel free to contact me if you have any questions. I didn't really talk about it as much as I should have, but mentoring was a very important part of getting me to where I am. And as much as I love mentoring, I also love being mentored. So I have many mentors, and I would encourage you to have many as well. And so please reach out to me if you have any questions about any part of this. And my website's kind of hard to find, so I put a link here. I don't know how easy that is to type in, but please visit my website or send me an email, and I'd be happy to talk more. So thank you so much. And I guess I'm happy to take questions in the Q&A and however you want to type them in. So, thank you.